Good evening, everybody. Welcome to this evening episode of Pursue, and this is Pursue 14N, which is Hematology, General and Fundamental. We are streaming live from the famous Tata Medical Center, Kolkata, and thus today we have a very interesting, very important topic. The topic of the day, the things which is really moving very hot, which is role of molecular biology. Molecular biology is almost replacing everything, and it is creeping into every science. And today we are looking into what is the role of molecular biology in the present day hematological diagnosis. To speak on that, we have somebody who is an international star. We have Dr. Colonel Deepak Kumar Mishra. He is an MBBS MD from the famous AFMC Pune with Post-Doctoral Fellowship in Hematology from AIIMS New Delhi. Senior consultant in the laboratory, Hematology and Molecular Pathology, and the director, Department of Laboratory Sciences, Tata Medical Center, Kolkata. He has been the guest speaker in multiple national and international conferences with more than 80 publications in national and international peer-reviewed journal. He has worked for more than 25 years in the armed forces. He is a gold medalist in MBBS, adjudged the best medical graduate in 1983 from VSS Medical College, Odisha. Then did his MD Pathology from AFMC Pune. His major area of interest have been flow cytometry, molecular genetics, in hemato-oncology, including MRD in acute and chronic leukemia, including MDS, MPNs, and multiple myeloma. He has also developed a special interest in solid tumor molecular pathology, including liquid biopsies. Trained in hematology in multiple centers in the West, UICC fellow and visiting faculty at the famous Mount Sinai, New York, King's College School of Medicine, London, Mass General Hospital, Boston, and Harvard Stem Cell Institute, Stanford Medical Center. And he has also been the fellow of the Indian College of Pathologists and Indian College of Hematologists, member National Academy of Medical Sciences, chair of the ICMR Task Force Subcommittee on MDS, and national convener of the Indian MDS Registry Initiative. Currently, he is the president of the Molecular Pathology of Association of India and the former president of IAPM, ISHBT, former secretary of IAPM, and former editor of the Indian Journal of Hematology and Blood Transfusion. Currently a board member in the board of directors of the International Society of Laboratory Hematology and the chair of the American Society of Clinical Pathology International Certification Program for India. Member of the EHA committee and coordinates the educational activity of the European Hematological Association. Who better than Dr. Mishra to talk about the role of molecular pathology in hematology? So I would request Dr. Mishra to start. Let me share his screen. Sir, can you see your screen?
Yes.
Yeah, please start.
Good evening to everybody, and my profound apologies for this technical glitch. I couldn't connect to my desktop, and somehow Nadeem could circumvent this problem, and I'm thankful to him. At the outset, I must thank Nadeem for putting in place this Pursue series of lectures, and I think they have immensely helped people in the virtual times, and people have been educated on the weekends. And today I'm going to speak on the role of molecular biology in the present day hematology diagnosis. Thanks for his humble introduction. I was thinking that the introduction is going to be more than my talk. Thank you, Nadeem. As you know, that molecular biology is going to be the future of diagnostics in all areas of pathology, be it liquid tumors, be it solid tumors, be it benign hematological disorders. The molecular biology is here to stay and is going to be the poster boy or the poster girl for the pathologists in years to come. The sooner the young pathologists they develop an interest in this particular area is the better. And I'm sure everybody, every MD student and every freshly passed out MD will heed this advice and will develop interest in this particular area. Could I have the next slide, please? This is Tata Medical Center in the night, and I bring greetings from the hospital. May I have the next slide, please? Could I have the next slide, please? Uh, this is a slide which I would like to start with. This is a seminal publication that happened in the journal Science way back in 1960, and it is a special article which gave you the Philadelphia chromosome, small little, almost like letter to the editor by Hungerford and Noel, this teacher and the student, and you can see that the Philadelphia chromosome, the karyogram is in the background, and since then we have come a long way from 1960 till today, and I will take you through the journey of what molecular biology has happened after this seminal discovery of Philadelphia chromosome. Could I have the next one, please? After this, which was quite, you can see that the chromosomes that you can see, which you saw in the background, was further characterized and refined. And today, the Jimsa banding has come a long way in giving you very, very clear bands, the light and dark bands. And here you can see the 922 balanced reciprocal translocation seen in chronic myeloid leukemia. And now this is the sine qua non of the diagnosis of CMLs in myeloproliferative neoplasms. And you don't diagnose CMLs without looking for the 922 translocation or the BCR-able transcripts by different techniques. After it was discovered, then you had the molecular characterization of this BCR-able. You saw that you could have two types of transcripts, the P210 and the P190, based on the weight of the transcript. It is P210, which is E13A2 and the E14A2, and P190 is the E1A2. Usually CMLs have P210, that's E13 and E14A2s, and the ALLs, particularly in adult ALLs, you have the P190. We devised various ways how we can detect it. We developed the reverse transcriptase PCR technique. May I have the next slide, please? We developed the RT-PCR technique where we could detect in a multiplex PCR. Next, please. It will show up the, yes. In the multiplex PCR, you could see the bands and the annotation. You can see that the different molecular weights could characterize E1A2, E14A2, and E13A2, along with the three controls, particularly the K562 cell line that gave you the BCR-able transcripts. May I have the next one, please? Then we developed the FISH probes, and this is a dual color, dual fusion probe. You can see that the fusion signal is there where you can find a little tiny bit of the yellow signal. That's the fusion transcript, and you have the normal red and the normal green, the BCR and the able. They are the housekeeping genes seen in every cell. Following that, it took almost 40 years for this poster boy in the Time magazine where we could see that there is a bullet against this particular transcript, and it's called the targeted therapy, and imatinib became the first targeted molecule to target chronic myeloid leukemia. It was a signal transduction inhibitor 571 developed by Novartis called Gleevec, and now you have almost 40 Indian generics in the market, and the cost of treatment per a month from 1.8 lakhs has come down to almost 5,000-6,000 rupees. So the treatment of CML has evolved over a period of time because of the targeted treatment. May I have the next one, please? I can show you that the concept of it is that whether you want to do sniper shooting or carpet bombing. When you carpet bomb, you don't know whether you will hit the target. Thousands of bombs, you may not hit it. But if you sniper shoot, possibly you will hit the target. And that is what is the fundamentals of targeted treatment in genomics. If you look at this molecule, you can see that the normal BCR signaling happens in a very, very interesting way. You have the BCR-able domain, and you have the ATP, you have the substrate, and the kinase domain, which activates the substrate protein, which is the PI3 kinase by a process of phosphorylation. And the activated substrate then initiates the signaling cascade, culminating in cell division and proliferation. You can see that how Could you move the cursor? Yes. How the ATP is binding in the ATP binding groove, and it is phosphorylating, and the signaling cascade is happening, and that's how the cell division and proliferation happens. Next, when it is abnormal, the mechanism of action is different. The imatinib mesylate exactly occupies the ATP binding groove, and that deactivates the signaling cascade. And as a result of that, because of the lack of signaling, it inhibits the proliferation and survival. You can see that imatinib mesylate is sitting on the ATP binding groove, and thereby the downstream signals doesn't take place. Next, please. When you have already targeted the BCR-ABL, it is important that you quantify as to how much you have targeted and how much is the residual leukemia. And by that, you want to quantify the residual disease. And I'm going back to almost 22 years ago. This is a slide which I borrowed from Hammersmith Hospital from Jaspal Khida, who used to be working in Professor John Goldman's laboratory. And he has developed this limiting dilution assay where you serially dilute the BCR-ABL transcripts and see that at what level the BCR-ABL dilution or the BCR-ABL signal vanishes. And it is it used to be very cumbersome, but that is how it started the quantification process of the residual disease. We have evolved a long way. The next slide will show you the screenshot of the real-time PCR. Here you can find that you are seeing the BCR-ABL transcript copies real-time in a real-time PCR, and you quantify it based on a standard curve. Next, please. So why it is important to quantify the BCR-ABL transcripts and how you quantify it is very important. Today, we have come to a stage where we can give the BCR-ABL copies in a standardized manner, because if you give the IS scale of BC, it is called the International Standard or IS scale. If you express the BCR-ABL copy numbers in IS scale, you align this BCR-ABL values to International Reporting Scale or IS scale. And that enables you to consistently interpret the results, it benefits the patients, it limits the variability of results, and it enables comparison of the response to treatment to various kinds of tyrosine kinase inhibitors in a consistent manner, and it enables patients to transfer from one place to the other without getting bothered about the BCR-ABL results. So when you talk about this International Reporting Scale or IS scale, you find that 0.1 is the limit of 3 log reduction when you treat patients of CML, and you want to achieve that in 12 months' time of treatment of TKIs. So if it is less than 0.1, then you are in major molecular remission. But to achieve that consistently, it requires a consistent laboratory. And because of that, you have, you see, the same patient, you have different points, but if you have the conversion factor to it, which is multiplied, just like you have prothrombin time, you have the International Sensitivity Index, and you express it as INR, International Normalized Ratio. You see, once you multiply that, everything will fall in line and you are at 0.1. So that's the concept, and whatever you may do, you'll always have variability in the result of BCR-ABL. The idea of having it in the international scale is to avoid this degree of variation that will affect the concordance of the major molecular response in chronic myeloid leukemia. Could I move to the next one? Yes. When you see this slide, where you can see that you are seeing the BCR-ABL transcripts at different logs, and the red line is the one that is the detectable limit in conventional cytogenetics, but you have to move beyond conventional cytogenetics. These days, nobody does a bone marrow to monitor patients of chronic myeloid leukemia. You do it from peripheral blood, and you do it from the RNA, you convert it to cDNA, and then you do the real-time PCR to find out what level it is. So when you start the treatment, usually you have about 10 to the power 12 number of cells, leukemic cells in the body. And when you treat at the point of morphologic remission, you have it at the level of 10 to the power 6. And when you have at molecular remission, you have it at the 10 to the power level 3. That's the major molecular remission. But today, we are going even deeper than 10 to the power 3 because
we have a concept of treatment-free remission in chronic myeloid leukemia. Patients have been on imatinib for years and years by now because it started in 2001. Now almost 20 years have passed, and many patients are in durable, complete, major molecular remission or even complete molecular remission to warrant or to think about stopping imatinib. So when you want to stop, you have to be in higher scale and you have to have a deeper response to imatinib to stop the treatment. So the monitoring of chronic myeloid leukemia by a deeper molecular response is extremely essential to put patients on stop imatinib trial. Next, please. So why I have started the talk by through chronic myeloid leukemia, because it is a prototype disease in hematology, and this is the first disease where you could find the target and you first found the drug against it. So this is the starting point of molecular biology of hematological disorders. So when you treat patients with TKIs, with imatinib mesylate, patients develop resistance at some point. Some of the patients, not all patients. So there are different mechanisms of the development of resistance. You have some BCR-ABL independent mechanisms and you have BCR-ABL dependent mechanisms. In the BCR-ABL independent mechanism, you have pharmacokinetics and oral bioavailability of the drug, which depends on the cytochrome oxidase enzyme system in the liver. You may have activation of alternative pathways of signaling, like the SRC kinase overexpression. Rather than TKI, now it becomes SRC kinase. You can have clonal evolution, like you can have BCR-ABL transcripts having multiple copies in a single cell. So clonal evolution can happen with other cytogenetics which are adverse to the patient, or you can have single nucleotide polymorphisms in the drug transporting mechanism by the development of OCT1 gene overexpression, where the drug enters into the cell and effluxes out very quickly. Because of that, it doesn't act on the cell. So that is also another mechanism of resistance. But when it comes to BCR-ABL dependent mechanism, you develop a lot of missense point mutations in the kinase domain, thereby the drug doesn't act. So 35 to 60% of the patients develop TKI resistance in chronic myeloid leukemia by an dependent mechanism on BCR-ABL kinase domain. Could I have the next one, please? So this is a cartoonized form of this resistance. You can find that you have a proliferating leukemic stem cell where that blinking red dot is actually the mutation that has happened. So what will happen in the next slide, just a tap, you can find that the imatinib mesylate cannot go and bind there in the ATP binding group and will come back, and what the substrate will go and bind and the proliferation will start. So thereby, when the resistance happens, the cell proliferation continues and the patient comes back to relapse. And you have to check the tyrosine kinase domain mutation in them. Next slide is our Tata Medical Center data of almost 8 years. We have studied almost 154 patients by now who are showing resistance to TKI by suboptimal response, and we found that almost 44 patients were positive, which is 28.7% of such patients to have a TKD mutation or the missense mutation. 90% of the mutations are missense mutations. They are point mutations, they are gain of function mutations, and we could detect that so that you can change the first line TKI of imatinib mesylate to either dasatinib or nilotinib or ponatinib, and the list is endless. And you can find that there are some which are resistant to one and sensitive to the other. So you have to check the chart, which one is sensitive and resistant to what mutations. Next. So I pay my gratitude to this man who is no more. He is Professor John Goldman, the professor and head of hematology at the Hammersmith Hospital, which used to be called the Royal Postgraduate Medical School in olden days. And he is a phenomenon in chronic myeloid leukemia, and world over he is known as a CML man. And I pay my little gratitude when I was in that institute on a UICC fellowship for chronic myeloid leukemia. Next slide, please. So now I come back to my laboratory and our laboratory. You can see that this is the nucleic acid extraction laboratory, which has both biosafety cabinets. We have a fully automated walkaway extraction platform from from QIAGEN called QIAsymphony, and we have all the infrastructure that is required to do RNA extraction, DNA extraction, genomic RNA DNA. We have the extraction of the cell-free DNA, the FFP DNA and RNA, and everything we can extract in this particular laboratory. Next slide, please. This is the frontal view of the QIAsymphony, the QIAGEN fully automated DNA RNA extraction platform. It can also do FFP RNA extraction. And when you extract the RNA or DNA, it is extremely essential that you check the quality and the quantity of the RNA and DNA. You have three platforms here. You have the nanodrop, you have the qubit, fluorometer, and you have the depth station. All three are available in our lab to see the quantity and the quality of the extracted RNA and DNA. Then you move to the PCR lab, and the PCR lab will have a lot of thermal cyclers. We have a host of them, almost 5-6 of them, along with the real-time platforms which can do the quantification of transcripts. Here you can see the Bio-Rad machine, the Verity is here, and you have the gel documentation platforms which can give you the conventional PCR and the various modifications thereof, including the RT-PCR, the nested RT-PCR, and all modifications of PCR can be done, including ASO-PCR. Everything is possible in that, and we have the gel documentation system from Bio-Rad, which will document it, and you can capture it on the screen for further study. Next, please. These are the three real-time PCRs. We have acquired the fourth one from Agilent, the Area Max, which is going to be installed soon. This is the ABI 7500. You have the Rotogene from QIAGEN, and you have the Quant Studio 3. And we're going to add the fifth one, that's the Quant Studio 6, in some months' time, which is in the ordering mode. Next, please. And this is the gel doc area. Next, please. This is the area where we do the sequencing. We have currently 5 sequencers, and the sequencer, that is, you can see the right top is the gene reader from QIAGEN, which we are returning because we are not very satisfied with its performance. Otherwise, we have 2 from Thermo, that's the Ion Torrent PGM, and the Ion Gene Studio 5, and we have the Illumina MySeq. So these are the 3 NGSs, and we have the ABI 7500, which is the 8 capillary Sanger sequencer, which you'll see in the next frame. Next, please. This is the ABI 3500 genetic analyzer, which is a 8 capillary DNA sequencer, and the other one is a ProFlex PCR machine, which can do 4 into 48 reactions at one time, different reactions. So these are the 3 sequencers: the Ion Torrent PGM, the Ion Gene Studio S5, and the Illumina MySeq, which is the bread and butter of next generation sequencing laboratory. Next. So next I show you some images which will give you an idea as to how you can utilize these infrastructure for diagnosis. You know that myeloproliferative neoplasm is very important, and you have JAK2 exon 12, JAK2 exon 14, you have the calreticulin, you have the CMPL. All these mutations are important to be detected to diagnose myeloproliferative neoplasms. And when you come to polycythemia vera, 99% of the patients should have the JAK2 V617F gain of function point mutation, and that you can detect by this allele-specific PCR. You can see the positive, negative, and the non-template control. And the left-hand side is actually the positive patient, which is positive for JAK2 V617F. So JAK2 V617F, we do it by 2 techniques in the lab. We do it by droplet digital PCR as well as by ASO PCR. And ASO PCR will have these bands, which you can see and can diagnose that, yes, this patient has with JAK2 positive. Next, please. When you talk about the other markers for chronic myeloproliferative neoplasms or myeloproliferative disease, you do the calreticulin mutation. You have calreticulin mutation of 2 types. One is a 52 base pair change, another one is a 5 base pair change. And this is the type 1 calreticulin mutation, which is the most commonly found mutation in MPNs. Next one is the type 2. Next, please. This is the 5 base pair change, And this is the FLT3 NPM1, where you can find that there's a FLT3 mutation being positive, which is a 181 base pair change from the mutant to the wild type allele. And FLT3 is extremely important to be detected in acute myeloid leukemias, because today you have an anti-FLT3 drug in the form of midostaurin and sorafenib, which is used along with the induction treatment in patients of AMLs. Next, and these are all pictures of fragment analysis which has been done in the Sanger sequencer. When you have a busy transplant, could I go back to the previous slide? When you have a busy transplant unit and you do allogeneic transplantation, it is very essential that you do chimerism analysis. Chimerism analysis is done by short tandem repeats. We have 16 STR kits which we use for the chimerism analysis. This is just one STR marker called penta-E, where you can see that there is a pre-transplant DNA, there is a donor DNA, and the recipient or the post-transplant DNA. You can find that the post-transplant is almost the same as the donor DNA. Thereby, we have a calculation and we express the degree of chimerism as 98, 95, or 100%. Here, this is 100% chimerism post-transplant in day 100. So next, please. So how important is molecular genetics to monitor patients of treatment? Diagnosis is extremely important. Here you see the electropherogram of the Sanger sequencer. You can see that you have mutation being present. This is the BRAF mutation in exon 15. You can see that BRAF is usually present in hairy cell leukemias. It can be present in melanomas. It can be present in papillary carcinoma of thyroid. So in multiple lesions you can detect. And here this is the forward primer, and you can see in the next, in the reverse primer, this is V600E. It is important that when you do Sanger sequencing, you always have a forward reaction and a reverse reaction. This is called bidirectional Sanger sequencing. It is important that you do bidirectional Sanger sequencing to avoid errors, because if you see a mutation, which you can see in this arrow, you can see that small peak which is inside the larger peak. That's the mutation where you can have, this is T2A, and in the reverse you will have A2T, and that is how you will say that yes, mutation is present. Sometimes you have stutter on the base, and these stutters are to be interpreted. If one of the runs is not having that peak, you say that this is a stutter and not a mutation. Next, please. This is the V600E. This is the reverse. The previous one was the forward. So this is V600E mutation. Then the technology of PCR is changing. Now we have digital PCR. Everything is digital these days. You know, analogs are all vintage. So they will be there to stay. But digital droplet PCR is catching up. We were the first institute in the country to have this in the diagnostic field. There are two units. One is a droplet generator, and another one is a droplet reader. And you'll see that our scientific officers are working on the digital droplet PCR, and I'll show you how it is beautifully utilized in the treatment of patients. Next. This is the machine from a closer view. One is a droplet generator, other one is a droplet reader. Next. So this is a plot where you can see that this is a promyelocytic leukemia post-treatment which has got a BCR1 transcript. As you know, promyelocytic leukemias have three types of transcripts: BCR1, BCR2, and BCR3. 98% of the transcripts are BCR1 and BCR3. Extremely rarely, about 2% you can have BCR2 transcripts. It is important that at diagnosis you must characterize what type of transcript the patient has. So a qualitative RT-PCR to detect the transcript is important for further analysis down the line when the patient is under treatment. So here you can see that these are the plots of the digital droplet PCR, where you have the controls, positive control, you have the sample, and you have the non-template control. And every drop is one reaction. This is almost like flow cytometry, where every single cell is depicted as a dot on the scatter plot. So you can find that. I will show you the plot in the next slide. Next, please. You can see that in this bar diagram, you can see that we have depicted the housekeeping gene of ABL, and we have shown you the PML transcript. This is the BCR1, and you can find that in the positive control you have this, and in the post-treatment, post-consolidation thing, you have zero transcripts of BCR of PML transcript. So this is a beautiful platform to do rare event detection. This is not so good as for diagnosis. You can use it for diagnosis, but this is a wonderful tool to use for rare event detection, like copy number detections in BCR-able. We are using it for P190 transcript detection in acute lymphoblastic leukemias. We are using it for acute promyelocytic leukemias. We are using it for detection of NPM1 mutated acute myeloid leukemias. We are using it for JAK2 mutations in myeloproliferative neoplasms. Next, please. So this is another plot where you have the BCR3. So same thing, you have positive control, you have sample, and you have non-template control. Next, please. And you can see this reading, and you can find out that there is an element of PML copies in the second bar, and that's how you know that the disease is still there in a very reduced scale. And what you express it as fractional abundance from the transcript to the mutant alleles. So I'll not go into the details of that. This is a slide that shows the workload, the molecular genetic samples processed in the lab. You can see that over the years, we are almost 10 years past, and I presented this in one of the presentations in the hospital only a week back. So we are increasing our numbers so far as the number of samples that are processed. Next, please. This is you can see that these are the total number of test parameters that we do, but we have rationalized it, and the numbers have reduced to about 45 or so. And 45 numbers are all types of tests, including solid tumors and liquid tumors. Next. Yes, this is you can see the road. The road is going up and up. It had a dip in 2020 because of the pandemic. And in 2021, we are almost catching up. By April, we have almost 1,400 some odd tests in molecular genetics. Next. This is a diagram that shows you what all we do. So this is an inverted pyramid. You can see that the commonest test that is performed in the laboratory is EGFR mutation. We see almost 750 to 800 new patients of lung cancer in a year. And we have done almost in 2020, which was a pandemic year, almost 600 plus tests in EGFR mutation. Then is the BCR-able, and then is many other tests that are there. And we will go to the other slide. Next, please. And in case of the molecular markers of hematolymphoid neoplasms, you have diagnostics, you have MRD, you have pharmacogenomics, and you have chimerism. We have AML, ALL. We see almost 1,800 new patients of leukemia in a year, all types. And everything is flow cytometry is done in all. Most cytogenetics is done in all. We have in-house cytogenetics. Mayur Pariyar is the consultant. We have one of the best cytogenetic laboratories in the country with almost 100 FISH markers, both for solid and liquid tumors, and conventional cytogenetics, including stress cytogenetics. So in MRD monitoring, we have the RQ-PCR for BCR-able. We have ddPCRs. I just told you the markers. And we can go up to 10 to the power minus 3 sensitivity for different markers. Like for AML, we have NPM1. We are now developing the WT1, that's the Wilms tumor 1 gene. We have the PML-RARA, the BCR-able, and now we are trying to detect the fusions like 821, the inversion 16s, also as laboratory developed tests. Pharmacogenomics is very important. TPMT mutation in ALL is extremely important because that will determine how much of 6-mercaptopurine you will be giving. So thiopurine methyltransferase is a very important mutation that determines how much of 6-MP the patient receives. Similarly, BCR-able kinase domain mutations, the FLT3 is important now because midostaurin is added to the induction upfront in all patients of AML induction. PML RARA is important because you will determine whether the patient goes for ultra-retinoic acid or arsenic trioxide in patients who are PML RARA positive. And the chimerism I have already described, that it is important to monitor patients post-allogenic transplants by doing the STR analysis in that. Next. This is the ELN guideline 2017, characterizing the acute myeloid leukemias to favorable, intermediate, and adverse risk. I will not go into the details. You have the highlighted things that are done in our laboratory. We do FLT3 NPM1, we do CBPA, the biallelic CBPA, we do the c-KIT mutation. You all know that 821 translocation is a favorable risk AML, but if you have c-KIT mutated in that, it becomes intermediate risk. So permutations and combinations of the presence and absence of these mutations will put acute myeloid leukemias into favorable, intermediate, and adverse risk. So before the physician starts the treatment, he knows which one to transplant, which one to treat with chemotherapy. So it is important that the patients go for transplant in the first remission in patients who are intermediate and adverse risk. So risk stratification of AML is important if you want to give standard of care treatment to patients of AML. Next. So this is again the pre-ALLs. You know that ALLs constitute almost 60% of the acute leukemias in our center. Pediatrics 75%, adult 25% of precursor BALLs. And out of the 25% precursor BALLs in adults, almost 40% of them have BCR-able transcripts in them. So Philadelphia positive adult ALL is very, very poor prognosis. In addition to that, we have a host of other markers, like conventional cytogenetics will tell you whether they are hyperdeployed or hyperdeployed, and whether they have IAMP amplification, IAM21, or they have the BH-like chromosomal status, like CRLF2 positive and other things. So we have a large number of FISH markers to characterize precursor BALL to its finality. Next. So this is the TALLs. TALLs have lesser number of chromosomal abnormality unlike the BLS, but it also has cytogenetics which can characterize them as poor, favorable, and intermediate. So you can see that they have been characterized, and we have a large number of markers to look for the risk stratification of TALLs. MLL gene translocation is one of the most common to give the adverse risk to TALLs. Next. So these are the things that we do in our center, in Tata Medical Center. For myeloproliferative neoplasms, we do next generation sequencing. We do the myeloproliferative neoplasm reflex panel that starts with JAK2. JAK2 has got two exons, exon 14 and exon 12. Exon 14 is the conventional JAK2 V617F. You have calreticulin, you have CMPL, and you have the JAK2 exon 12 in case of polycythemia vera phenotype. For MDS, we do the comprehensive myeloid gene panel. I'll show you that in the next slide. CML, I have already told what we do. And AML, I have said that we do comprehensive myeloid gene panel along with the AML panels in case the patient cannot afford NGS. I'll show you the panels. And APMLs, you know that we do RT-PCR, ddPCR for PML RARA. NPM1 and FLT3 in all, and in FLT3 we give you the FLT3 allelic ratio. And as you know that if you have the mutant to wild type allelic ratio of more than 0.55, it is adverse prognosis. And we have the other translocations, which we do it by nested PCR. We do the c-KIT mutation in case you have the 821 translocation to put a favorable risk AML into the intermediate risk acute myeloid leukemia. Next. In addition, we have ALL panels. We have the ETV6 RUNX1, that is the 1221, TCF3 PVX1, that's 119, and the KMT2A, that is the MLL gene translocations. On lymphomas, we have the clonality assay. We do the IGH and TCRG clonalities in case of different difficult lymphomas where you are confronted with something which could be a reactive lymph node versus a lymphomatous lymph node. So this is a routine test that we do almost every week. TP53 has gained a lot of importance because anything which has got TP53 is poor prognostic. So we look it in CLLs and we look it in many solid tumors and we look it in the germline setting also for Leigh-Fraumeni syndrome. MYD88 along with the CXCR4 for lymphoplasmacytic lymphomas and DLBCLs, BRAF V600E for hairy cell leukemias are standard tests in the laboratory. TPMT is routine for all acute lymphoblastic leukemias, particularly in pediatrics, where 6MP is included in the maintenance phase of the treatment, which they receive for almost 2.5 years. Next. I mentioned about comprehensive Myelogen panel. I mentioned about AML panel. So we have two AML panels, 1 and 2. We have the FLT3 NPM1 and the biallelic CBPA. And in the panel 2, we have the IDH1 and 2 and the NRAS KRAS. Why I have said that we have two panels. The panel 2 is gaining importance because today you have anti-IDH and anti-KRAS drugs available as targeted treatment. So they are all in Sanger sequencing platform, but it is done in cases where the patients are relapsed refractory AMLs, where we want to give some second line or third line treatment. Next. This is what is the comprehensive Myelogen panel, which is a 69 gene panel, where you have fusions and mutations in the same study. So here we do the RNA extraction of the genomic RNA extraction, we do genomic DNA extraction, then we convert the RNA to cDNA and we pull it, and then we have about 21 hotspots, 19 full genes and 29 fusions in one study. So it is very, very useful because in one study you will get the status of fusions. You do not have to send it for cytogenetics because fusions are detected and we have validated it and 100% concordance with cytogenetics FISH. So hotspots are there for 21 genes and full genes like SLX1, which is present in MDS, BEAC or CAL reticulin. There are so many, the list is very vast. ETV6, all are full genes. And this is a wonderful panel and our turnaround time is 2 weeks. And this is a test which is replacing the conventional Sanger sequencing for acute myeloid leukemias and MDS and MPNs. So all myeloid malignancies we do this. Next. So this is our first 100 patients of comprehensive Myelogen panel. You can see what are the most common mutations that we have found in myeloid malignancy. So that is the bar diagram which can show you what all we have seen in AMLs. Next. Not only AMLs, but all myeloid malignancies, including MPNs and MDS. So these are the comparative features of next generation sequencer versus NGS versus Sanger sequencer. As you know, that Sanger sequencer is the gold standard of sequencing, but gradually it is getting replaced by NGS. And here in the Sanger sequencer, you do the dideoxy chain termination method. You detect the ATGCs. I showed you a few of the electropherograms of Sanger sequencer. So here we have low throughput, low sensitivity, slow speed, and you only uniplex it. Whereas in NGS, you multiplex it, high throughput, high sensitivity, but you are deceased with the turnaround time because the time of run is longer. So your turnaround time will be longer. So we pool the sample because it is expensive. So we run it for all assays at least once in 14 days, once in 2 weeks, to be 10 working days to be precise. Whereas Sanger sequencer can be run every day. Next. So this is actually the method of AmpliSeq technology. I will not go into the details because many of you may not go into the technicalities of it. So there is target enrichment, you have multiplexing up to 24,000 primer pairs. We don't do 24,000, but it can multiplex large number of genes, like you just saw 69 genes could be multiplexed. So it requires DNA and RNA in very low quantity, 10 nanograms per microliter is good enough for this assay, whereas you need higher quantity for Sanger sequencer. The key implications are it focuses on specific hotspots. You have two types of assays. You have a hotspot assay, where you are looking for the hotspot mutations, or you can have whole exome sequencing. Whole genome sequencing is not possible with the platforms that we have in the lab. You need either a NextSeq or a NovaSeq or a still higher 10x genomics. So the conventional diagnostic next generation sequencing is possible in the three platforms that we have, and we are making the best use of them next week. So I mentioned about the next generation sequencing. It is very, very important that you have good quality RNA and DNA, both genomic DNA RNA and also FFP DNA RNA. Right now we are standardizing the Oncomine Focus assay for solid tumors, particularly for lung cancer. It will give you both mutations and fusions. All these MET mutations and all these exon 14 skipping mutations, everything can be detected in that. So in that you have to have very good quality FFP RNA and DNA, and it is possible only to detect that by this nanodrop qubit fluorometer and by the TEP station, which is the next one. Next. So this is the TEP station, which also gives rise to the DNA index and the RNA index. And next slide, please. You can see the plot. This is how it gives the DINs, and this is how you will reject or accept the samples based on the quality of DNA, so that you don't waste next generation sequencing cost by putting bad quality DNAs into it. So sometimes we get blocks from outside the hospital for reviews, and many times because of poor fixation, because of the processing, we don't get good quality DNA, and the DNA is very shredded, and many times it fails the standards for putting into the NGS. This is in nutshell the workflow in NGS. I will not go into the details. You have the RNA convert to cDNA. Then you have the DNA on the other side. So the FFP DNA and FFP cDNA gets pooled. And then you run the assay in a single multiplex assay. And that green red is actually the loading of the chip. And you have quality standards as to how a chip is acceptable so far as the run characteristics are concerned. So this is the Iron Gene Studio S5. So that's the latest one that we have. We had the Iron Torrent PGM, and then we added this about a year back, and this has increased our throughput as well as our assay capabilities manifold, because this is the latest in the range of thermo products. Basic principle remains the same. High throughput sequencing, 2 million to 130 million reads per run. This is very high. Next. This is how the technology works. Is basically it releases the proton or ion, which changes the pH in the well, and that's how it detects the different nucleotides, A, T, G, C. Next. So this is the kinds of interpretation that you have. You have two types of interpretation when you get a NGS report. You have the analytical interpretation and you have clinical interpretation. Analytical interpretation remains within the laboratory. That is because we need to fulfill the analytical characteristics before we report it clinically. The clinical assessment, as you must have seen in case of NGS, you have tiers, tier 1, tier 2, tier 3 mutations. Tier 1 is the pathogenic mutation, which is reported in multiple databases. And what you are more important and more concerned about is VUS, is called variant of unknown significance, which is likely pathogenic, non-pathogenic. And I'll come to that in the next slide. Next, please. So here you have these mutations either in the form of a diagnostic biomarker, prognostic biomarker, or you can have it as therapeutic biomarkers, because you have a lot of targets now currently available. So today you have knowledge bases, databases, which can give you annotated results telling that these mutations will work with these targets. and these drugs. Next. So this is what it is. You have the values, the SIFT values, which is less than 0. This is something called in silico prediction. So when you have a mutation which is not reported anywhere in literature, so you do a in silico prediction whether this mutation is pathogenic or non-pathogenic, and you have the SIFT score, and based on that you can say that likely pathogenic, non-pathogenic, or pathogenic. Next. But to conclusively say that this is pathogenic, you have to do functional studies, which is not possible in our laboratory. This is actually done in research labs. Next, please. And these are the various databases. Now, please go back to the previous slide. These are the different databases. The ones that we use mostly is Cosmic database, the ClinVar, and the HGVS database. These are all databases which are in the public domain. You can look for whether the mutation that you have found is already reported or it is already annotated as pathogenic, non-pathogenic, or likely pathogenic. These are the platforms that I have already showed you, and the current recent one that we have acquired is the Illumina MySec, which is very study equipment and is the platform of choice in most research laboratories. And gradually the Illumina realized that they are poor in diagnostic. So now they are propagating Illumina MySec as one of the diagnostic platforms. So we are one of the labs which acquired them for our diagnostic work, and we are currently planning or standardizing our Oncomind Focus assay and the HRR gene assay, which is very important now for CA ovary and others, in this platform. Next. So this is a comparison of direct Sanger sequencing or bidirectional Sanger sequencing and NGS. I have already mentioned few of them, so I'll not go into the details to save time. Next. So the difference between real-time PCR and Sanger sequencing, real-time PCR will only detect what you are trying to detect. Sanger sequencing is also same, but there only it is qualitative, not quantitative, but the real-time PCR, it can be both qualitative and quantitative, very short turnaround time, same day reporting, and very little expertise is required to run it and hardly any computational need. Whereas Sanger sequencing, you will need a reference sequence to compare, and you have to study the forward primers and the reverse primer runs for analysis. Next. The next one is the digital PCR. I told you, the digital droplet PCR from Biorad, and the real time. Real time will need, this is a relative quantification because you need a housekeeping gene. And digital droplet PCR is absolute quantification because you don't need a housekeeping or a reference gene for reporting. So this is absolute quantification, that is relative quantification against a housekeeping. Next. This is the molecular markers that I have already mentioned. May I have the next slide, please? I think this is a repetition. Next. We have already covered this. Next slide, please. Next. In any molecular, then I'll go back. In any molecular lab, it is very important. No, next, the one that came on the, yes. So in any molecular lab, quality control is extremely important. So our lab subscribes to the UK NEQUAS for many assays, including the VCR-able quantification, the PML-RARA quantification, the FLT3 NPM1 chimerism, TKD mutation, the flow cytometry, anything we do, we always subscribe to this UK NEQUAS, and you can see here that this is a program for, I'm not able to read this. So where we are satisfactory, you can see that the result is satisfactory. We get 2 samples every quarter, and we are passed in that. Next is another UK NEQUAS sample. You can see that we are passed in this as well. The next one. So the idea of showing this is that when you have a molecular lab, it is very important to have confidence in what you are doing. And these things give you the confidence. When you get an external sample, you run it, send your result, and you pass, that gives you the confidence that what you are doing in the lab is correct and the best. And there is always scope for improvement. Sometimes we have the dot falling on the outlier, means it may have a high positive or negative bias, and we take care of that so that we are right in the center. Next, please. So this is our molecular team. We have three consultants, myself, Mayur, and Sushant, who was our fellow before, had joined us as a consultant, and we have two fellows at any given time in molecular genetics, and we have a host of scientific officers, all are MSCs and some are PhDs in the molecular lab. And human resource is extremely important. So this is just not a clinical pathology lab doing urine and stool. This is a molecular lab dealing with DNA and RNA. So you need highly qualified people who are cerebral, not working at the brainstem level. So they have to think, they have to review the literature, they have to produce results, analyze, and produce papers and publications at the end of it. Next. So these are our teaching and training programs. We have the Post-Doctoral Fellowship program in molecular genetics in our hospital. Two positions are there. There are 2 years program, extendable to a third year provided the candidate wants it and we find them suitable. We also have training programs for DRNB in hematology, because we have two vacancies. So right now we are into the third year, so 6 people are undergoing DNB training in hematology. And then we conduct annual workshops. Last year we couldn't do it. It's a weekly program. Regular weekly interlab training programs. We attend all DMDs, disease management groups. And we are planning to have an exclusive molecular disease management group, or molecular MDT. So that should be there in another month's time, training academic sessions for DMLT students. And we have a host of, all the time we have visitor observers from various institutes in the department as part of regular training activities. Next. We encourage our fellows and the staff to go and present in international meets, like Sushant, when he was a fellow, presented in Salt Lake City in the American molecular pathology meeting. It was the best paper award in Berlin. He attended in the European AMP. Then a lot of other scientific officers have attended and fellows have attended the international meets and have presented papers. We do encourage them to do that. Next. We send people for international training. Dr. Kallol Sir, he has completed his PhD, went to University of Chicago for a training. And we sent Visoja Paul to Hammersmith Hospital to learn the NGS technique of tyrosine kinase domain mutation as a UICC fellow. Then we sent Poonam Santra for King's College London training in NGS and RT-PCR. So we do send people for overseas trainings so that they do good quality work. With that, I thank everybody for a patient hearing. So what I would like to propose, Nadeem, that this is a basic fundamental presentation. So I would like to make a molecular diagnostic case-based presentation, how molecular diagnostics have helped in the diagnosis of real-life patients. So as and when he finds time in anything, I would love to do that. And now I end the topic and I'm open to questions. Thank you.
Thank you so much, sir. Wonderful presentation, so detailed out. Definitely, we would like to have the case-based presentation. That would be very important for everybody to learn from there. At least we have got a fairly good idea about, role of molecular biology in hematopathology and the extent of work which is being done in Tata Medical Center as one of the leading centers in molecular pathology in the country and maybe in the Southeast Asia. Wonderful job, wonderful presentation, and definitely I will get in touch with you and we'll try to fix it up so that we have a big talk on the case-based presentation and how exactly in practice it is being done and how it is affecting treatment and protocols. Just let me see if there are any questions on YouTube, and then I can do that. Right. There are no questions, sir. Thank you so much. Thank you so much
May
for
I
coming.
ask a question?
Yes, please.
Deepak sir, yes. Sir, I'm Dr. Gayatri Gogoi from Assam Medical College. Recently, sir, you visited our college and there was a lecture by
Yes, I was there.
Cancer Care Foundation. So we would like to hear from you. Maybe your next lecture I like to propose on CML, BCR-ABL detection by PCR, and MRD, because that is the commonest one that we can practice.
The bread and butter.
Yeah, that's the bread and butter, correct. Yeah, we will definitely try to put that up, as early as possible.
So I just
Thank you.
touched upon, but not in that great detail, but we can have an exclusive presentation on CML diagnosis and monitoring.
Yeah,
Right, sir. And
that would be a good one.
one little bit in detail so that we can actually practically do it here. We have real-time PCR. We have one a medical research unit sponsored by ICMR. So we can actually do it here.
I have
So
told
practically to that. Right, sir.
that when you people are ready, we will develop your lab, no problem.
Right, sir. Thank you so much, sir. And maybe we will send one of our junior who is very interested in molecular pathology, especially hematology. Yes, sir.
I have promised your principal that.
Right, sir.
The principal was there in that meeting. I did promise them that we will train your people. Yes.
Right, sir. Great, sir. I mean, I was waiting to listen to your this presentation. Since Nadim posted regarding this talk.
In spite of the technical glitches, you are still hanging on. My thanks to you.
No, thank you so much, sir. I have been listening your presentation, international, national conferences.
Thank you. Thank you very much.
You're welcome, sir.
Right. Thank you, sir. So we have 2 lectures lined up for you. Great. Yeah, we'll definitely fix up a time when you're.
I acknowledge that you are doing great service in this times organizing these. So at least we are trying to refresh our knowledge as well as we are trying to speak something in the virtual mode.
Right.
It's extremely important. Yeah.
Right. Right, so thank you so much. Thank you everybody for joining in, sir. Thank you so much again. God bless you. Thank you. Right.
Thank
Good
you.
night. Good night, everybody.